Hello, AP Biology. Once again, let's do some more notes. Today, we are going to talk about proteins and amino acids, which are my favorite things ever because they're the coolest. So let's dive right in. I kind of say everything's my favorite. I'm going to be honest with biology. There are times I really mean it. Okay, let's talk about amino acids. I made a small adjustment to the notes. You'll probably notice it right away, and I'm going to update them. For biological use, there's a ton. Oh my gosh, protein. There's a reason I love protein so much is because they literally do anything and everything. If you need something done, a protein will probably do it. They are enzymes, which are things that little biological machines that make things happen. They can be defensive. You can store stuff, like store energy in them. You can transport things with proteins. You can make transport like across membranes and stuff. They can be hormones. They can be on the surfaces of cells as receptors. They can make things move. They are what muscles are made of, and other things. They can be structural, like collagen. They're super awesome. Actually, I'm going to show you. There's a part of your the page in your book that I want you to see that I think sums this up gorgeously. Maybe I'll take a picture of this as well and I'll put it in there. This is page 78. 78. And there's this beautiful table here. And look at all the things that proteins can do. Isn't that amazing? It's so cool. Obviously, proteins are going to come up a lot. They're very important. Proteins are also great because they are encoded for in DNA. This is what DNA does. DNA becomes proteins. So it kind of makes sense that they do all the stuff. It's kind of funny, actually, when biologists back in like the 30s were trying to figure out how, like, what was the macromolecule that actually that inheritance came through. They were kind of thinking it could have been DNA or proteins. And a lot of scientists were actually betting on proteins because proteins are way more complicated. DNA is kind of weirdly simple. So they thought, well, yeah, way more information can be encoded in proteins. They were wrong, of course. The fact that DNA is so simple is what makes it so freaking amazing and useful as for encoding. But it's because what they can create is an astounding array of proteins across all different species. It is absolutely insane and beautiful and wonderful. And it's why I love biology so freaking much. So let's do this. Diving right in. Back to the notes. So definitely check that out. You might even want to make a note of that off on the side. I did leave a lot of space in your notes for that. Okay, so proteins are made up of amino acids. We also call proteins peptides. So if you see the word peptide, you know that we're talking about proteins. A peptide, or an amino acid, whichever one you want to call it, has three parts. It has an amino group. Amino, to me, whenever I think of amino or amides or ammonia, anything like that, nitrogen. It's the word that comes into my head. So on one end, you have a nitrogen group. Then you have a carbon. And attached to that carbon on one side is a hydrogen. And on the other side is an R group or a side chain. We call them R groups. It's kind of like X in math. So it just means something goes there. It could be as simple as another hydrogen, or it could be something extremely complex. The R groups are super important because that determines the properties of this particular peptide. The final part is the carboxyl group. Carboxyl is referring to this carbon double bonded to an oxygen and single bonded to another oxygen, which then has a hydrogen on it. Sometimes it doesn't. Sometimes there's a hydrogen is missing here and there's an extra one here. That way it has a charge on either side, but it's not really a big deal. So those are the three parts of an amino acid. When you put them together, you call it a polypeptide because it's made up of more than one peptide. Yes. So the bond is also called a peptide bond. So what will happen is let me see, actually, hold on. Do I have a picture of a polypeptide? I don't think I do. I don't have a specific picture of that bond. Oh, wait, there was one earlier. I'm just going to backtrack a second because we got to find that bond. Okay, the bond happens between, of course, it's a dehydration synthesis because they're all our dehydration synthesis. So it happens between the nitrogen of one group and the carbon of the next. So if we're going back here, this OH would pop off. One of these hydrogens would also pop off, and that's how the bond happens. The dehydration, HOH, H2O, dehydration synthesis. There you go. So the pattern you'll always see is nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That is the main string of beads for that. Okay, R groups. R groups are super great. There are different kinds of amino acids. For the purposes of biology, there's about 20 essential amino acids. These are ones that some of them we as human beings can make on our own, others we can't. So when you say that you have to eat protein, we're talking about amino acids because some amino acids, if we don't eat them, we cannot make them ourselves. So we have to have them in our diets. Otherwise, it's very bad. I don't remember which ones they are, but there's a whole bunch of them. It might be some of these, but I think we can make some of them on our own. I'll have to look it up. Or you can look it up. Google. It's great. Okay. Generally speaking, they come in a couple of different like general flavors. You don't have to know all of this, but some of them are polar and charged. So if you'll notice, these ones have charges to them. There's like little pluses there. There's some minuses there. This one, I don't really know where the positive is, but it's somewhere. It's probably with that no, it's not with the nitrogen. I don't know. Some of them is they're charged. If they're charged, they're definitely polar and they're hydrophilic. So these ones are also negatively charged. They're going to be polar and hydrophilic. So that is going to have an impact on the overall structure. So if you, like, for example, one of the big things that happens, they'll ask you on the AP test, and it's just kind of important just to know in general. If you change one amino acid to a different amino acid, it's going to make a structural difference in the overall protein, which is going to have an impact on its function. If you switch out two amino acids that are very similar to each other, the change might be very subtle. So for example, if you switch out an aspartic acid with a glutamic acid, this one's just a little bit longer. It's one extra carbon. So it's going to make a difference, but they have similar properties. So it's probably going to be relatively minor. If you switch out side chains, these R groups that have completely different properties, it's going to make a much bigger difference. It also depends where in the protein it is. That's a whole other thing. We'll talk more about enzymes and proteins later. Okay, some of these are polar but uncharged. So I'll give you a little hint in terms of knowing if something is polar or not. If you see a lot of oxygens and nitrogens, it's probably polar. Oxygen and nitrogen make things polar. If it doesn't have a lot of oxygens or nitrogens, it's probably not polar. These are some examples of hydrophobic or non-polar side chains. So if you'll notice, these side chains, especially near the ends, there's mostly just carbons. I've mentioned this before, but I'll mention it again. Anytime you see these lines, there's invisible carbons. And there's also hydrogens that go with those carbons. So like, for example, here on leucine, this little end here is a carbon. There's a carbon here, there's a carbon there, and there, and there, and there. So there's actually 1, 2, 3, 4, 5, 6 carbons. And each of these carbons would have some hydrogens on them. So there would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens. It's not important. You don't need to know it. It's just good for you to know. Organic chemistry stuff. It's a whole thing. You can learn that later. Okay, so those are the hydrophobic ones. The special cases, there's a couple of weird ones over here. They shouldn't come up too often. Two of them, I'm just going to mention these because I love them because they're like my favorite. Methionine and cysteine cysteine are really interesting because they have sulfur. You'll notice none of these have phosphorus. So you do not need phosphorus to have proteins. Good to note. You might need sulfur though. Sulfur, especially cysteine, is really interesting because if you have two sulfurs on a polypeptide chain, they are some of the few molecules that can create a covalent bond within that structure. They're called disulfide bridges because they create a bridge because two sulfurs like to bond to each other. I mentioned this. I'm going to come back to it. Yeah, I'm going to come back to it. And if I don't in the video, remind me in class. It has to do with beta carotene and maybe alpha carotene. Let me double check. I may have to double check this. Hair keratin. Sorry, one section. Just checking. I can never remember if this is true or not. Yes. Cysteine, yeah, absolutely. Okay, I'll double check a few things about it. Anyway, those are our groups. Again, the big lesson I need you to get out of this is that they determine structure, which helps determine function, and that if you switch them out, like if there is a, like if DNA gets messed with, what is that called when it gets it's the word I'm looking for. If you get like irradiation and then there's some sort of mistake in DNA, you can end up with the wrong protein or the wrong nucleotide, or not nucleotide, the wrong amino acid or peptide, and that can mess with your structure of a protein. Okay. Yes, so polypeptides, covalently bonded, and specifically peptide bonds. I'm going to just add that in here real quick. Peptide bonds. This is something that you'll want to try to remember if you can. Okay, so proteins are super awesome. It's all based on the R groups, and it's in a sequence, and any given protein can be made up of as many polypeptides as it wants. Some are very simple. They're just made of one polypeptide. Some protein complexes are made up of like a dozen individual polypeptides that all come together to do something. So there are 4 structures. We divide up the structures of proteins into 4 levels of structure. So we've got the primary, secondary, tertiary, and quaternary structure. And there is a beautiful, here's a lovely little diagram that helps explain it. And your book also has some of these as well. Oh yeah, I'll show you. This is on page 82 and 83. And let me just show this. I'm going to give you a share of this just because it's a really delightful picture. I think it's very, very good. This is why I like this book. Okay, so I'm going to use this to explain it. All right, your primary structure is literally the linear chain of amino acids. So it's the order. So if you notice here, there's always a 3-letter code to help remember all their names. And a protein can be super, super long or very short. This is actually not a very long protein. In reality, the chain would be way, way, way bigger. You'll also notice there's always, like I said, an amino end and a carboxyl end. So there's a directionality to this. So that's just the straight chain. So just the order. You can sequence proteins just like you sequence DNA. There are machines that do that. So you could get a protein sequence, which is just the linear chain. This is what the protein's made up of. As soon as this protein starts to, is pumped out as part of translation from the ribosomes, it immediately starts folding in on each other in different ways. All of those side groups interact with each other. So for example, hydrophobic side chains are going to try to avoid water. So they tend to kind of tilt inwards and create little pockets to stay away from water because they're hydrophobic. Hydrophilic parts will more likely to spread on the outside. So the placement of those will help fold the protein into the correct configuration, whatever that configuration is for whatever purpose it is. Okay, The overall three-dimensional structure, once this whole thing is put together, the overall three-dimensional structure of the polypeptide chain is actually called the tertiary structure. Okay, so the tertiary structure is the major three-dimensional shape. It's determined by all of those side chains, just like whatever interaction is most stable. The secondary structure is patterns and motifs that you'll see within that. So it's recognizable patterns. There are two main patterns that you need to know. There is the alpha helix. So that's kind of it's a helix, just like DNA. So it's just like little spirals. There's always most of the interactions that keep this three-dimensional structure in place are hydrogen bonding. That is super important. Again, there are a few exceptions of covalent bonds, like the disulfide bridges that I mentioned. So yes, there's an alpha helix. And another similar pattern is this beta pleated sheet. So what that really looks like in a larger molecule, I think this diagram really shows it very nice. Okay, you can see here, this is a helix, an alpha helix. And then this region here and this region here are beta pleated sheets. So they're kind of motifs within that interaction. Yeah, so let's see. They mentioned here the hydrophobic interaction. So hydrophobic interactions will force that part of the molecule inside to be surrounded by more hydrophilic interactions. Oh, this actually does mention disulfide bridges. Lovely. It is between two cysteines. Let's see if you can see. Yeah, perfect. Okay, so you can see here, this is also really great. So here's a hydrogen bond. So that's a place where things are kind of close together. Here's a hydrophilic interaction. They call it van der Waals. It's also known as dispersion forces, but that kind of holds them together. And this is a disulfide bridge. And you can see it's an actual chemical bond that really holds it together. A really good example of this is keratin. So keratin is the protein that makes up your hair and nails. And keratin is very rich in cysteine. And what that does is it's why like hair and nails and even rhinoceros horns are made of keratin, like hooves of different animals are made of keratin. And they are so strong because of those disulfide bonds. So actually, this is crazy. If you ever get like a perm or relaxers or any sort of like chemical treatment to change hair texture is determined by the placement of those disulfide bridges. So if you have curly hair, I have kind of like medium curly hair, it's because of the placement of those disulfide bridges. So if you go to get your hair, either a perm to make it curlier or a perm or a relaxer to make it straight, like permanently, it's the chemical process is essentially breaking these disulfide bridges, restructuring the hair into the new position you want it in, and reforming those disulfide bridges. Like, that's the use for them. Which is why the new hair that grows in isn't altered, but just the hair that you had is pretty much permanently chemically altered, because it's not just like hydrogen bonds, which could kind of reshape themselves. It's covalent bonds, which are really hard to break and reform. So it's pretty cool. There you go. Oh, you can also see an example of an ionic bond. So an ionic bond is happening here because there's a positive and a negative, a fully charged positive and a fully negative side chains, R groups, which are really attracted to each other. So overall, this is the strongest interaction because it's a covalent bond. This is probably the next strongest because it's ionic. That's a really strong interaction. This is the third. It's not quite as strong as this, but still pretty strong. And this is definitely the weakest. But depending on where they are, it'll hold the position of that protein in place pretty well. It's pretty cool stuff. That's a good picture. I'm glad I found that one. Very, very informative in a lot of different ways. I just love it so much. I just like want to look at it. Okay, we'll move on. Oh, one other thing. This is where pH can start to mess with this. So if you start adding more hydrogen ions, that lowers the pH and makes it more acidic. Those hydrogen ions are going to come in here and start messing with this. and it's going to start potentially messing with this, and it will definitely start messing with these bonds. This bond, because it's covalent, it's less likely to get messed with, but it could. But these two, it would be super easy for those hydrogen ions to come in and compete for interaction and make these attractions or bonds much weaker. And that could really mess up the structure of your protein. It could start to unravel. And that would be very bad if you want it to be in a particular shape for a particular function. So there you go. Possibly a reason why adding, if you put a lot of acid in your hair, it may not be a good choice all the time. So there you go. Okay, quaternary structure, let's do that. All right, quaternary is what happens if you take multiple polypeptides, take those tertiary structures and just moosh them together. So there's a lot of examples of these. This is a transthyretin protein, which I don't know what transthyretin does, but transthyretin is made up of 4 identical polypeptides that all come together to form one big complex, and that's the quaternary structure. They've got another delightful example here. Collagen is made up of 3 polypeptides that all come together. They're also all identical, and they all twist together. Hemoglobin's another great one. Hemoglobin is made up of 4 total, they call them subunits. That's a good thing to know. So collagen has 3 identical subunits. They're different colors just so you can see them, but they're all the same. All alpha helices. Nice. Whereas this other one, primarily, mostly beta pleated sheets. Okay, final thing, hemoglobin. Hemoglobin is made up of 2 different types of polypeptides. There's the alpha and the beta subunits. So the purple ones are alpha, the blue ones are beta. You get 2 of each. 4 of them total create the hemoglobin protein structure, which is insanely important because that's how we deliver oxygen. It also has, there's a heme group, which is what actually holds the iron. If we let just iron ions just freely running through your body, it would just be a disaster. So it has to be very tightly held. Actually, when I was in college, I took a class called Bio-Inorganic Chemistry, which is a lot because there's biochemistry, there's inorganic chemistry, and there's organic chemistry. Those are like 3 different sub branches of chemistry. This combined all 3. And it was all about these types of groups. So it's like, how do you take an inorganic atom like iron and put it in a biological system? So it was a really cool class, and we definitely learned a lot about those groups. It's called a coenzyme. It's a whole thing. Okay, anyway, so that is that. I'm just going to double check and see if there's anything super exciting on this page. Yeah, so I'll give you this quick example. This is a classic example. If you start messing with the structure, it will mess up proteins. So this is a great example of that. The B subunit of hemoglobin, one small substitution from glutamine to valine takes the whole structure of that B subunit in hemoglobin and changes it completely, which is the cause of sickle cell anemia. So one small alteration can really change everything and takes it from a normal, perfectly functioning red blood cell to a not really very functional blood cell at all, which can cause people lots of problems. So that is, again, a connection of how the structure determines the function. So there you go. Okay. There's more stuff we can talk about here. I do like this real quick, just about denaturation and renaturation. So when you denature a protein, that's what it means to kind of mess up that structure. It is denatured. It's taken out of its natural formation and messed up. Usually you can use heat or chemicals, various chemical treatments like changing the pH will denature a protein. Definitely heat will. If you turn up the temperature, proteins will just wiggle apart into spaghetti. Okay, I'm gonna go back to the notes. I get so distracted so easily just because I love this stuff so much. All right. That's actually kind of all I have to say about proteins. There is probably more. I mean, there's always more, but we're going to study it in more detail. I think that's the most basic stuff that you really need to know for right now. So in terms of proteins, let's see if we have any other resources that might be helpful to you. Share screen, go to Teams. Okay, let's look at Unit 1 Chemistry Overdive. Okay, for notes, obviously these notes. There are some notes on, I think it would be under carbon. That could be useful. Let's see if we have classwork or assignments. If you want to learn more about proteins, functional groups for sure. This worksheet will definitely be helpful there. Functional groups are, like our groups are known as functional groups. The different nucleotides also have functional groups. That's like the adenine, the thymine, the glutamine and cytosine. The G and this, the ATGC of the nucleotides. Those are functional groups. The parts that actually do the thing. Yeah. Macromolecules overview might have something. Oh, protein structure. There is a POGL on it, so you could definitely check that out. Let's just see what that looks like real quick, if it'll load. This could be quite interesting. Yeah, check it out. That looks pretty cool. Heck yeah, I'm liking this already. This looks, look at that. There's diagrams. It's beautiful. Beta pleated sheets, alpha double helix. There's a tertiary structure. There's a quaternary structure. There's 3 different things all combining together with lots of beautiful questions. Okay, so that could definitely be worth a look at if you want. I like POGLs a lot. Okay, let's see about labs. No labs. No labs. Oh, for labs, I forgot to mention this earlier, but I'll mention it now just for funsies. This one, the oil and water would be really good for the beginning of the unit. But you could throw it in anywhere. It's just kind of talking about hydrophobic versus hydrophilic interactions, intermolecular forces, that kind of thing. So, okay. Awesome. I think that's good for now. I'm going to do another video on carbohydrates and one more on lipids, and then that'll be good for all of this. So there you have it. My lighting has gotten worse because I'm relying on this overhead lighting, but there you go. All right. Until next time, toodles.